absolute honor to be recognized with this prestigious award, which of course is in the honor of late Shri Manohar Parrikar ji. If there is one thing which I would want the students here to take home after about 30 minutes is that all these challenges that you heard about, and I'll present an example from our own work as well, is that all these challenges need solutions across different disciplines. No one single discipline, no one single expertise, no one single individual is going to be able to actually manage all these different challenges. So no matter what your current background might be, what your current interest might be in science, you still could learn other fields, learn other disciplines, and more importantly, also, instead of reinventing the wheel, synergize your efforts with other colleagues, other experts across different disciplines, and then you could have outcomes which are emergent problems. So what we have been working on is thinking about cancer as a complex adaptive system. Now, this is a book I read in summer 2012, just before starting my PhD. And this book was sufficient to motivate me to work in the field of cancer further. It's a wonderful popular science book. If many of students who have not read it, I would recommend you to read this. It documents the history of cancer over multiple centuries, when it was first observed, what are the current treatment strategies, what across different civilizations, across different societies, and cancer is still called as the emperor of all maladies because we have not yet been able to completely tackle this disease. Now, what is cancer? Cancer is not one disease. Cancer is actually a group of diseases with two generic properties. One is that cells grow in an uncontrolled manner. In normal scenarios, every cell in the body has a mandate to not grow after a certain number of times. When that mandate is disrespected due to various perturbations that happen in the system, that is when we get cancer. And another property that many cancer cells have is that they no longer remain restricted to where it is. It initiated in the body, but actually spreads to other different organs of the body as well. Now, this is a property which other diseases usually do not have. If it's a heart disease, it will stay in the heart. If it's a lung disease, it will stay in the lung. It will not directly go and lodge itself in different parts of the body and begin to disrupt the functions of the body. So here is an example. These are this is the liver, and what you see as patches here are pancreatic cancer cells which have moved to the liver. Now, how does this movement happen? This movement largely happens through blood vessels, which are spread all over the body. So cancer cells in one organ would enter the circulatory system, travel all over the body, keep making exits at different organs of the body, and can also grow at other parts of the body. Now, this is just some data in terms of prevalence. So this is how the number of cases have been increasing in the United States over the past 50 years. Number of projected cancer cases also larger. So this is about 26 million cases projected in the next 15 years or so. Also, number of cases in India have been growing, and next year at least 15 lakh cases are expected to show up. Now, what is also worth noting is that despite the increasing number of cases in the US, what you do not see is a large increase in the number of deaths that happen due to cancer. So this is still hovering around 6 lakh deaths per year, 600,000 deaths. But in India, we already have more deaths than that despite having small number of cases. Now, this could be due to multiple reasons. The diagnosis is later, the treatment options are limited, we are perhaps not able to afford all treatments, so on and so forth. There's a large list of cases, but clearly it is no longer a US problem. This is impacting our country day in and day out. And this speaks for itself. As per a recent report, 1 in 9 individuals in India are likely to develop cancer throughout their lifetime, right? So there is a large population of students sitting here. 1 in every row is going to perhaps develop cancer eventually. That is the statistics that we have right now. So what are the current treatment therapies? We already got introduced to radiotherapy earlier by Dr. Kapurkar. So the first therapy, the first set of therapies, which was pioneered much earlier, is of course surgery, where you basically excise the part of the organ which has tumor. When X-rays were discovered, then of course came radiation therapy around the turn of the century, around World War II, also discovery of chemotherapy. Now all these therapies, as you can see, are more generic in nature. You have to localize where to give the therapy, and that is how this works. But in the 1980s, we started having what is called targeted therapy, which means that now this particular set of molecules is going to bind to specific molecules which are present predominantly on cancer cells, and that is why this is more specific, this is more targeted. And around 15 years ago, we also started having immunotherapy in the clinic, where the general idea is that you do not directly target the cancer cell, you actually over-enthuse or over-activate your immune system and say, please go kill the cancer cells which are around. Right? This also got the Nobel Prize in 2018. Now, if we have such diverse options over a period of a century and a half that you just saw, why have we not been able to conquer this disease? And there are multiple reasons behind it, which I'll try to highlight. So let's look at what are the different stages of cancer progression. So initially, when tumor initiates in one or more cells of the body, then it begins to grow. Because it grows, these cells grow much faster as compared to normal cells. They begin to disrupt the structure of the organ, and it no longer remains restricted, but it actually spreads to other parts of the body, a process called as metastasis. Now the time scale that you see here depends on which cancer we are talking about, which treatment it was given, etc. But what I want to highlight here is the acceleration in the process that happens once it starts to spread. Once it starts to spread, then of course the time that a patient has goes down dramatically, because multiple organs are being impacted, and that leads to multiple organ failure eventually. So what is the progress that we have made as a community in the past 5 to 6 decades? We now have much better tests, going back to also the first talk, much better tests to diagnose the disease earlier. So that's a technological advancement. And we also have more molecular understanding of what genes are involved in accelerating or deaccelerating cancer. And again, going back to the concepts of lifestyle, what lifestyle factors may increase the rate of getting cancer. But as you would notice, all these particular steps largely restrict themselves to the first stages of cancer progression. We have not made any major breakthroughs once in terms of curing metastasis, in terms of restricting cancer once it begins to spread. And that is where our research currently focuses on. Metastasis remains the cause of 90% or more cancer-related deaths. Now, when I started working into thinking about these questions, one thing which was very surprising to me was that if you think about the most common type of cancers, such as prostate cancer, lung cancer, liver cancer, breast cancer, these cells do not have the intrinsic property to move. All these organs, these are not red blood cells which will traverse throughout the body. These are specific organs in a specific geometry, in a specific organization, specific structure, and the function of each of these cells is to maintain that. Right? Now, how do these cells then begin to move? How do they know where to go? How do they settle in a completely different environment, biochemically, biophysically, etc.? Those are the kinds of questions we have been looking at. Now, what are the challenges in developing drugs against metastasis? Right? How many of you students sitting here have seen any parts of Harry Potter movie series? Excellent. So the first scene that you see here is close to the first scene of the first movie, right? And this scene that you see here is close to the last scene of the last movie. If I were to give you only these two scenes, these two images, and ask you, tell me what happened in between. You have not seen the movies, you just are given these two scenes. Just based on these two images, we will not be able to say anything conclusively about what happened in between. You don't even know if the people in scene 1 are the same as people in scene 2. This is where metastasis research is right now. You have the primary tumor, which is where cancer originated. You have metastatic tumor. Once a patient has developed metastasis, you can biopsy it and get an image. You have no clue whether it's the same cells who reach. There are new kinds of cells that originated after therapy. Therapy might also give rise to other questions. Right? So these are all open questions. The dynamics is very, very poorly understood. When and why do cells leave? Who forces cells to leave? What is the criteria? Why do they go? How do they decide where to go? Do they go as single cells? Do they go as clusters? All these are open questions. We have some answers, of course, but not to the stage that we can begin to develop therapies. Which organs do they stop by? And how much time it takes for them to grow clinically notable metastasis? So in stark contrast to the previous talk that you heard, where we have detailed quantitative information about how a chain reaction is going to proceed. You can control the amount of power being generated and use that precise calculation for developing plants, power plants. We have very limited quantitative understanding of how exactly this happens, and without numbers you cannot make accurate predictions. There's another unsolved challenge in cancer treatment, which is drug tumor relapse. Right? So even if it is not metastasis, cancer can come back at the same organ. And here is an example. This is melanoma, which is skin cancer. So this is pre-treatment. That's why you see all these lesions or all these spots present in this patient. 15 weeks after therapy, you notice that these lesions have largely disappeared, but only 8 weeks later, in presence of the therapy being continued, cancer comes back. Okay? Just 8 weeks, just 2 months. What goes on here is very, again, largely speaking, very poorly understood. Are there pre-existing resistant cells? There were some cells which just did not respond to this therapy that was given, but they're pre-existing and the number was small enough so that they could not grow a lesion, but once given an opportunity, they again blow up. Or does the treatment itself gives rise to new kinds of cellular behaviors which then allow the cancer to adapt further, and so on and so forth. Now let's think about the dynamics of cancer metastasis a little bit more quantitatively. So I'll have a few questions, and it will be very much appreciated if students can participate in this. So the first question is, let's say 10,000 cells leave a primary tumor. Primary tumor is where the cancer originates. How many of them will complete metastasis? Any guesses? How many of you think that it will be 50% or more cells which will form metastasis? Our body is of the order of 10 to the power 14, 10 to the power 15 cells. Let's say 10 to the power 4 cells, 10,000 cells leave a primary tumor together or at different points. How many cells will complete metastasis? How many of you think it will be 50% or more? 10% to 50%. 1 to 10%. Less than 1%. The correct answer is 0.01%. 1 in 10,000 cells which leaves primary tumor is able to form metastasis on average. It is such an inefficient process, yet we still don't know how to target them. Right? Imagine what would have been the scenario if this were a more efficient process. Okay? So these experiments are of course done. You inject cells into the bloodstream in mice, and then you see over as a function of time how many cells eventually grow tumors. And of course in this experiment you already injected the cells in the bloodstream. So you skipped one more step, which is the part of getting into the bloodstream, which would reduce the efficiency even further. Okay? Now, the journey of metastasis is, if you think from the perspective of a cancer cell, is a very challenging journey. Right? Think about, a cancer cell, individual cancer cell, which leaves their home organ, traverses the bloodstream, where the environment is constantly changing, where the immune system is trying to attack it, where the drugs that you're being given, the patient is being given in the clinic is trying to attack it, so on and so forth. And the oxygen levels are changing, nutrient, everything is changing very, very dynamically. How do cells adapt, right? How? So one fundamental property which helps the cells or us as individuals succeed when we traverse through changing environments is adaptability. How fast we are able to sense and respond to what is happening to us, right? And this adaptability, again, this is, you could take it any particular context. Also, as Charles Darwin alluded, the species that is most adaptable to change is the one that is going to be strongest. Because fitness of a population, of an individual, be it, again, going back to the talk on longevity, be it the fitness of an individual, be it the fitness of a society, it is going to depend on the environment. You cannot define fitness without an environment, right? So, and when you talk about environment, you think about adaptability to that environment. Right? Next question. how many of you think that every cancer cell is equally adaptable? So 1 in 10 power 4 is able to do that. How many of you think this answer is yes? Every cancer cell is equally adaptable. Okay, so everyone thinks it's no. But that's the correct answer. But how do we figure this out? What experiments one would need to do? Because in that, so if every cell is equally adaptable, probably it's even more difficult scenario. Thankfully, some cells are less adaptable. So all we need to do is understand the ones which are more adaptable and then go ahead and try to reduce their adaptability. Right? So these kinds of experiments were done where, again, I'll, without going into technical details, I'll give a sort of analogy, is that if you try to figure out what is the, is every student in class equally good? The answer is clearly no. How do you figure it out? You basically segregate the class into, at the end of the day, one single student who is not able to interact with everyone else, and give them the same exam, and then see what is the distribution of marks. Right? That's what happens in exams. Exact similar experiments were done in cancer in mice, and they showed that some cancer cells could grow many more metastases and some could grow much less metastases. So clearly every cell is not equally adaptable as well. So what we know so far is that cancer cells are heterogeneous. They have different personalities, and this is the technical term for this is called phenotypic heterogeneity. So heterogeneity means variability and phenotype means the behavior. And we also know that cancer cells can be adaptable. They can change their identity with time. A cancer cell which is more efficient in using oxygen at time X might be less efficient in using oxygen in time X plus 1 because it lost this property and it tried to gain some other property which was more important for it to survive in the new environment that has come up. Right? So clearly these properties of adaptability or plasticity is the technical term for it, and heterogeneity are playing a crucial role in relapse and drug resistance. And these are the kinds of questions that we'll be looking at. Now let's go back to all therapies that we talked about. Surgery, radiation therapy, targeted therapy, chemotherapy, immunotherapy. All of them have some assumptions. Okay, and I'll try to highlight those assumptions and the impact of those assumptions. So let's say that we were able to completely profile a tumor. Now there is advanced technologies, you can measure every single molecule with very high resolution, right? So let's say you say, I'm going to take the tumor of a patient and measure whatever different molecules I want to measure and say there are 4 different kinds of cells present in a tumor. And I also have targeted therapy for all 4 of them. We are not there yet as a community, but let's say that even if we reach this stage 10 years from now, what are the challenges? First of all, we are assuming that every red cell in this schematic is going to respond identically to the given drug and will have one and only one outcome, which is cell death. That's clearly not the case. You can do these experiments in the lab, you can grow cells, cancer cells in the lab, you can give whatever drug you want, whatever concentration you want. It is never going to happen that every single cell dies. Right? The other property, the other assumption we are making is that no drug is able to induce the transition of one red cell to become a blue cell. Right? What is drug? Drug at the end of the day is a stress. Stress can induce changes in behavior. We are not assuming that. We are also not considering that cells might actually talk to each other. Cells might cooperate with each other. There is ample evidence of this in the literature that cancer behaves as an ecosystem. There is an ecology. There are some cells which do half the job and the other cells do the other job. So again, the fitness is not defined at a single cell, but at a population level. Now, when you take all these assumptions together, what you reduce cancer to is a static, non-adaptive system. But in reality, it is a dynamic adaptive system. And a point to make here is, let's go back around 25 years when anti-angiogenesis therapies were being tried out. What is angiogenesis? Angiogenesis, as the name suggests, is the growth of new blood vessels. Cancer cells can grow new blood vessels so that they can get access to oxygen and nutrients. So the idea was, let's block that growth and we can cure cancer. These drugs were developed, clinical trials were started, and in a subset of patients who received these drugs, they started dying faster than patients who did not receive this drug. It was the exact opposite outcome. Now we know one of the reasons why that was happening was that in a subset of patients, these drugs were inducing metastasis. Because if you deprive cancer cells of what they need to grow, they'll figure it out a different way. They'll just go to different parts of the body. At the end of the day, these are our own cells, right? You might have heard of liver as the most regenerative organ in the body. You can chop off a significant proportion of the liver and it grows back. Why? Because there's adaptability in the system. That's how it has been designed. So are the cancer cells. This is not some infection. This is our own cells which have stopped following some rules. So we need to respect that these are adaptive systems and not dumb cells which are waiting for a signal from us to die. Now, how can mathematical models help, right? So think of any complex system, say weather prediction, right? At the end of the day, it's a prediction. And we reasonably trust these predictions. Why? Because you have curated these models extensively over generations, so that you have taken the data, you have understood basic, ideal gas laws, Newton laws, and so on, and then use that model to constantly match the new data being collected so that you reach an iterative stage where we are right now. Can we reach this stage of this prediction in cancer treatment? Maybe a few decades down the line is the goal. So what can mathematical models do? They can predict what is the possible dynamics given a set of interactions, how different factors such as therapy is going to influence that, and how can you eventually control that dynamics. So can we develop a mathematical model? Let's just present this one example from our work to study metastasis and drug resistance. Quick question again. When did you last develop a mathematical model? Students sitting here, how many of you developed any mathematical model in past 1 week? Okay. 1 year. Never. So all of you have never developed any mathematical models. I'm going to convince you that's not true. How many of you play any of these games or have played any of these games? You are all writing down mathematical equations in your head. You are just not solving them explicitly, right? You are guessing where exactly the ball is going to come, which direction you want to hit it, what acceleration, what velocity, where you want to park it, where is the fielder, what areas you are trying to avoid. It's all Newton's laws of motion at the end of the day. You can add friction and other complicated things in between, right? So we all use these tools consciously or subconsciously. It's basically a mathematical model is a formal logical way of thinking about a set of events and how they impact each other. At the end of the day, that's all that it is to do. Of course, there are tools. People who are driving, you have to guess whether these many seconds are left, you'll be able to cross the signal or not. S is equals to ut plus half at square. That's it. That's the simple equation that we're using there to calculate these kinds of things. Right? Stock markets. All mathematical models being used in multiple dimensions of our life, whether we realize them or not. So the question we started asking is, can metastasis and drug resistance drive each other? And the reason for this was this clinical observation that tamoxifen, which is a targeted therapy given to breast cancer patients, 1/3 of women treated with tamoxifen for 5 years come back with the disease within 15 years, and 1/3 of them actually have metastasis. So now you have metastasis and drug resistance happening simultaneously. Why? How? These were the questions that we were interested in, right? So this was a clinical observation. Now, just to give a glimpse of what exactly happens in the context of breast cancer treatment. So breast cancer, let's say a biopsy is done, it goes to a pathologist, and a pathologist measures these 3 molecules, which do some detailed functions in terms of cell growth. Let's not go into the technical details. But based on this, then a patient gets classified into A, B, C, or D, and those categories have very different treatment strategies. So if the classification is wrong, then of course the treatment strategies are wrong, right? So the most common ones is in this first category called estrogen receptor positive. Estrogen receptor, as the name suggests, is a molecule present on the surface of cancer cell which binds to estrogen, which is the growth hormone present in the environment. So the estrogen drives cell growth. Now, so that is why in a cancer cell, there is more number of estrogen receptors as compared to a normal cell. A normal cell knows how many times to divide, how many times not to divide. Cancer cells disobeys that, so there are many, many more molecules present on the surface. So the idea here, which is target therapy, which is for this particular subtype of cancer, is that it is going to be much more effective against these cells with minimal damage done to these other kinds of cells. Now, what are different ways in which tamoxifen cannot respond? Patients cannot respond to tamoxifen. So there are multiple ways, right? So how is tamoxifen working? Tamoxifen is binding to these molecules, and that disallows the binding of estrogen and going into the cell. So there are multiple ways. Not every cell might have very high levels of this molecule. This molecule might be present in different conformations, so the drug is not able to bind. And of course, this signal is not the only way cells divide. There can be many, many other ways as well. Right? So also in the context of metastasis, what we know, as I mentioned earlier, that most common cancers do not have the intrinsic property to move. So these are what are called as epithelial cells in our body, which line all the different organs. And then there are mesenchymal cells in the body, such as red blood cells, who actually travel all over the body. Now what is very well established in the field is that when epithelial cells in the tumor want to enter circulatory system, they need to loosen their cell-cell adhesion, and they need to gain the property to move. And this is a transition called as epithelial to mesenchymal transition, or EMT in short. And once they reach the other organ, they no longer need to migrate, they no longer need to invade, they need to settle down again, so they undergo the exact opposite function, NET. Now, what is the relationship between EMT and estrogen receptors, if any? One is driving metastasis, one is driving drug resistance. So what we know from literature is that, for example, there are these two molecules. This molecule binds to tamoxifen, this molecule does not bind to tamoxifen. These are the two different types of estrogen receptor. And also, this molecule can actually shut down the production of the other molecule through some detailed biochemistry, which I'm not going into the details of. So when this molecule is high, this molecule is low. What can also happen is that this molecule can actually induce EMT. So these particular molecules drive EMT, so they make the cells potentially more metastatic. And this is a feedback loop. This molecule then shuts down the production of this molecule. Now, when you see these feedback loops, you could notice that there are two possible states. This molecule is high, this is low, and this is low. So this is your primary tumor condition, when cells are not moving and cells are sensitive to tamoxifen. You can have the exact opposite state, when this molecule is low, this is high, and therefore this is low. Right? So you can have two cell states. In technical terms, it's a bistable system. You can have two cell states, and cells could switch back and forth between these two states. So the treatment of a drug could actually switch the cells. Now, each of these links that I highlighted here was known individually in the literature. What we have done here is to integrate that what are the consequences if you have all of these interactions going on together, and you begin to see non-intuitive predictions being made that you can have two cell states, right? This is like, say during cooking, you might have different ingredients, but unless you put them together in a specific sequence, in a specific amount, for a specific time, you do not get the flavor, you do not get the taste, right? So all the ingredients were known, but what we have done is the cooking part, right? We have integrated them both and used mathematical models to see what the emergent outcomes of these properties are. So we have shown that cancer cells can switch their cell states back and forth. They can be resistant or sensitive, epithelial or mesenchymal. And interestingly, this is the most interesting part, is that when cells begin to move, they already are drug resistant. So when they are exiting the primary tumor, the way these molecules connect to each other, as also validated by publicly available experimental data from these studies, and predicted by our model, that when cells are beginning to move, they may already be drug resistant. And interestingly, when cells gain resistance to drug, they also begin to move. So think of the clinical implications of this. Patients who are receiving this drug, which is 75% of the most common breast cancer cases, of course some cells are dying, but cells which are not dying can be more metastatic than what they were prior to the treatment of the drug. So the drug treatment is enabling new behaviors, which in this case is actually potentially worsening the situation. So now we are thinking about what combinations of therapies can be given to such patients, and these tests are being done in our experimental collaborators lab to see whether this holds out, and then we eventually plan to move to the clinical trials and so on. So what are the lessons? And I presented one case study, but there are multiple such case studies we and others have done so far. So you might think that you have a very accurate therapy, right? You are going to kill every single cell, because it's not chemotherapy, radiation therapy, which are more generic. This is targeted therapy, right? But there are some cells which survive. There are some cells which adapt. And cell death is therefore not the only inevitable output of any therapy. and they may induce a more aggressive behavior as well. And what we are now proceeding on is to use these mathematical models to then come up with better combination of therapies which can be taken further. So clearly these are open questions. All I have shown is a possibility that this can happen, and we have seen this in multiple cancers, multiple therapies, even immunotherapy we have seen this. But what we do not know is what percentage of cells die, what percentage of cells do not die. How is that percentage controlled by previous treatments, by the drug concentration, by the genetic background of the patient, etc., etc. Clearly, those are all open questions for which we would need more advanced mathematical models, more experimental data, and most importantly, more collaboration between mathematical models, experimentalists, and clinicians to take further. So I'll summarize what I have learned so far in this journey. So I started as an undergrad at IIT Kanpur, went on for my PhD, was a postdoc working with a clinician, and have been running my own research group for the past 6 years or so. So currently my research group contains students, undergraduates, postgraduates, PhD students, postdocs, from all different backgrounds. They are engineers, some of them are engineers, some are physicists, some are mathematicians, some are biologists. We also have ecology colleagues working together. And also we collaborate extensively with other colleagues across different universities around the world who also come from diverse different disciplines, because no one discipline is going to be able to solve what we solve.